for that introduction, Robert. So I've never given a talk on WebEx before, so hopefully you will excuse the awkwardness. I don't know, perhaps you can tell me, is my little video box visible with my talking head or not?
We see you talking, and there is this rectangle which it's still there. Yeah.
Can you see my title slide?
Machine learning for genomic of experimental evolution. Yes.
Great. Okay. Thank you so much. All right. So I'm going to assume that from henceforth things are working unless you tell me otherwise. Well, I think I'm giving this talk around a year late, because I believe I was originally scheduled to speak in Vienna in around April or May of 2020. Basically with this talk. So this will be less scenic for me, but hopefully the content will be conveyed adequately despite the vagaries of this linkage. I should say that the number of people who've contributed to the work that I'm going to describe is in the hundreds. The number of people who could reasonably be listed as authors on this talk would be around a dozen. However, I'm highlighting the four of us most guilty. I should also explain right from the outset that my chief role in the body of work I'm going to be showing you was as an enthusiast and a provocateur, in that in 2012 I basically conceived of this project and set especially Larry Muller, my primary colleague over the last 30 years, to come up with solutions for problems that I posed, greatly abetted by two of our graduate students, now graduated, Dr. Thomas Barter and Dr. James Kizos. And all this work was done at UC Irvine. So this is a diagram you're actually going to see again, but it's just up here to make the point that we use laboratory selection to create new populations, in fact, microcosms of populations, which of course will be very familiar to you at the University of Vienna. And it's also a bit of a cue to point out that whenever I'm talking about data, from here on in, I'm talking about data in fruit flies, Drosophila, and melanic aster. And I know I'm often guilty of failing to mention that that's our experimental organism. But one way to think of what we do when we do experiments in the field I like to call experimental evolution is we create new problems for us to study. However, they're new problems in a relatively defined microcosmic context, rather than the titanic diversity and complexity of populations living in the wild. And it's always been my belief since the 1970s, when I was a graduate student, that nature in the wild was hellaciously complicated, and therefore we needed to study something simpler, which has been my lifelong project. Now, complexity was, of course, greatly exacerbated starting in 2010 with the advent of pooled sequencing of experimentally evolved populations. And here I'm showing one of Christian's diagrams to basically outline the protocol of pool-seq or evolve and resequence, or whatever we choose to call it, the project by which we take a number of individuals, our favorite number is about 200, from a population and characterize their genomic variation with respect to single nucleotide polymorphisms, transposable element polymorphisms, structural variant polymorphisms, and even gene duplication polymorphisms. This could be done in two main ways. The way we've done it Irvine has mostly been to analyze terminal populations after they've evolved for some time. In Vienna, you'd like to do it sequencing from the start and then sequencing along the way. And I think we all have to agree that your approach is better, but unfortunately many of our populations at Irvine were created long before the advent of full sequencing technology. Well, here we come to today's problem, which is understanding gene-phenotype relationships, what Lewontin defined as the G to P map. And this is the problem of determining the genomic sites underlying the differentiation of individual phenotypes. And this is an important problem for our field because it's impossible to change just one biological variable in the course of experimental evolution. Instead, our protocols characteristically change many characters over the course of experimental evolution populations. And just to give a hint of what's to come, our basic approach to solving this problem is to use stepwise regression techniques, which of course feature a minimization of square deviations. And we're going to be talking about the addition of penalty functions in passing ridge regression and primarily the Lasso technique to shrink the variable complexity problem that we're dealing with. So the next 10 to 15 minutes are going to be about statistical learning or machine learning, or what the popular press calls artificial intelligence, a term that always makes me amuse because I think of it more as artificially diligent stupidity applied to problems that humans are not stupid enough to take on repeatedly. But it's just my point of view. Today we'll just call it machine learning. And there are two really important numbers to think about when thinking about this problem, especially because they don't have the normal relationship that you find in conventional statistical analysis such as we did in the 20th century. Those two numbers are little p, which in this context means the number of genomic features used as predictors across the entire genome. And generically, that number is on the order of 1 million. That might be 2 or 3 million in the case of human genomes, which are large and deeply characterized. It might be much smaller for less than genome-wide sequencing analyses. And the other number to think about is little n, which is the sample size. In GWAS, little n is the number of individuals you've sequenced genome-wide. In the context of experimental evolution, however, the level of independent variables that we need to keep track of is actually the number of populations, because we're doing pool-seq. So in effect, however we are characterizing our genome-wide sequencing, whether in terms of SNPs or transposable elements, we really only have as many independent data points as we have replicated populations. If you want to study a problem where p is much greater than n, there are no global optimal solutions for analysis, such as characterized conventional analysis of variance or multivariate linear regression, or any of the other what I call GLM, or generalized linear model, techniques that we used to use back when I was a young person. So we have to look at the modern generation of machine learning techniques to cope with our p greater than n problem. And fundamentally, what we're trying to do is we're trying to simplify our genomic models. And there are different terms that are used for this simplification process. One is subset selection, where the attempt is to reduce the number of genomic feature predictor variables that you're using prior to doing your linear regression. Another technique is shrinkage, in which perhaps we keep all the variables that we start with, but we shrink the magnitude by which they're allowed to contribute. And then a related technique is dimension reduction, where we're creating linear combinations of genomic features and going to a subspace of smaller dimensionality. And the classic example of this is principal components analysis. And I won't be using principal components analysis. Today I'll be talking about subset selection and shrinkage. So subset selection is our main focus. So if you were to just consider characterization of the genome at 20 sites, so p is equal to 20, and you attempted to derive the optimal statistics for characterizing populations, where you're looking at 20 genomic features, you'd have to iteratively examine 1 million alternative formulations, or 1 million alternative models. And even though there are shortcuts that have been developed, especially for least squares models, they still become computationally explosive or unmanageable. So today, I'm only going to be talking about stepwise selection, where you're going to be adding variables one at a time, which is computationally vastly more efficient. And the specific technique we're mostly going to be talking about today is FLAM, which stands for Fuse Lasso Additive Models. Only published relatively recently, 5 years ago, by Peterson and colleagues from Stanford. FLAM is a modeling approach which approximates potentially nonlinear effects of different nucleotide polymorphism frequency values across the 0 to 1 interval, and maps the impact of those different frequency values onto the phenotype using step functions. I'm going to show you a picture of these step functions. Then across all genomic sites, or in this slide called locus, the contributions are added. And the key feature of both the step functions estimated per locus and all of the loci that you add to your model stepwise is that there are penalty functions which penalize the gratuitous addition of steps in your step function per locus and additional loci as you go through your entire genome worth of data. But to just zoom back out for a moment, FLAM is designed to produce a sparse set of loci in order to predict the population phenotype. And this is a completely general tool, by the way, for going from gigantic sets of predictor variables to the characterization of a subset phenotypes. One thing I should point out right now, and I'll try to reiterate this point as we go through, is that the effort here is really focused on going backward. That is to say, once we have a FLAM predictor equation, our most interesting scientific result is, well, which loci did FLAM decide were the loci to use to characterize the phenotype? It may be opaque right now, but hopefully I can make that clear as we go on. So in common with all of these types of generic linear regression models, a central feature of these models is minimizing the residual sum of squares, which is the deviation of the observed data from the expected data derived from the formula. And So right at the top, it says the lasso can set some predictor coefficients to 0. So that's the most important feature today, is discarding information. And a key feature of the numerical technology is the use of what mathematicians like to call an L1 norm instead of an L2 norm. An L1 norm is simply the absolute value. So the L norm of 1 and minus 1 both equal 1, and so on through all real values. And an L2 norm is a square. Per locus, characteristically, what a lasso calculation will do is minimize the residual sum of squares associated with the genetic variation predicting the phenotype, plus lambda times a penalty, which is the sum of the absolute values of the beta parameters you use in your linear regression model, where this is just like your standard y equals mu plus beta times x, where x is your predictor variable. And there's, I know there's some relatively obscure math in the bottom 2 bullet points, but there's a relatively elegant way to view this problem geometrically. So if you're more of a graphical or spatial thinker, I think this is the most elegant way to explain the difference between the 2 main alternative stepwise forward linear regression analyses that machine learning can do. On the right, we have an example of a ridge regression machine learning, where we're just dealing with a 2-parameter space. Of course, this is standing in for n dimensions. I just got dumped out of my, let's see if I can, okay. Hopefully that glitch didn't mess things up for you, Aaron. So on the right is more of a standard way of thinking, and this is the way I tend to think, which is that you're trying to minimize your residual sum of squares, and equal valued RSS curves are shown in red. Those are the ellipses. So think of Laplace's model of the solar system. And roughly speaking, when you minimize the residual sum of squares subject to a penalty associated with having larger valued betas, you're attempting to minimize, attempting to find the point where one of those ellipses touches the blue circle. And at that point, so in this particular graph, it's at a point where most of the regression is due to the second predictor variable x2 with its associated regression parameter beta2. But there's a little bit of a contribution from predictor variable x1 with its regression parameter beta1. That's because the ridge estimators are minimizing the squared values of the beta parameters. Lasso fitting is shown on the left, and it's a completely different kind of problem in applied mathematics. Because you're using the absolute value instead of squared values, the minimization problem, geometrically speaking, is the approach of those ellipses to a spiky object defined by the absolute values. I'm going to show this again algebraically in a bit. But if you think in terms of n dimensions, you are dealing with a submanifold, an n-1 manifold, which defines the minimization of the residual sum of squares, being hooked on a very spiky n-dimensional object. And that spikiness enables us to include some variables and exclude other variables. So that's for those of you who are like me and like to think in terms of geometrical topology. Now let's go back to a more algebraic formulation. And The per locus machine learning minimization problem is shown in the upper left-hand equation, which is basically on the p minus theta sub j term, minimizing the deviations of the observed phenotype from a step function defined by thetas. And the step functions are shown in an example graph on the right, where you have the phenotype versus SNP frequencies. And those aren't the frequencies of three different SNPs, X1, X2, and X3. Those are three different values for the SNP frequency in a hypothetical experimentally evolving population, where the model that the Lasso machine learning came up with says basically that over the SNP frequency range X1 to X2, the model predicts the phenotypic value will be theta 1, and over the SNP frequency values X2 to X3, the predicted phenotypic value is theta 3, and there's this transition at the value X2. The penalty function is, to go back to the upper left-hand part of the slide, is weighted by lambda, a penalty function weighting term. So for example, if lambda is 0, there's no penalty for making the thetas as finely spaced as you like, so that you can approximate any type of nonlinear curve if lambda is 0. But lambda generically is greater than 0 in these kinds of applications. The D function, which is a matrix linear operator, basically penalizes you for each step that you take where the values are different. So for example, if the contribution of this particular SNP value to the prediction of the phenotype is uniform, there is no step taken and the penalty function goes to 0. So crudely speaking, if I can just put this into words for you, this machine learning tool has the ability, in principle, to model any type of relationship between SNP value and phenotype in a population, just looking one locus at a time, or one genomic feature at a time. Where that genomic feature could be a SNP value, frequency value, a transposable element frequency value, and so on. And but of course, this tool has to be adapted to an arbitrary number of loci. And basically what you're seeing here is one complicated line of algebra, which I'm going to again try to convert into words. There's a little bit more parameterization because now we're doing arbitrarily many loci. The top part of the equation is something you've already seen. It's basically the minimization of the residual sum of squares of the model relative to the phenotypes. If you go down one line, this is the multi-locus version with the first term. On the second line is the multi-locus version of the penalty function. There's both a individual locus penalty function and an overall loci penalty function. Put into words, basically, the more loci you add to your model, the more you get penalized for doing that. The fewer loci you use in your FLAM calculations to optimally predict your phenotype, the more FLAM likes that. To be very concrete about this, if you applied FLAM to populations which were differed only at one Mendelian locus of large effect, and you had lots of populations and lots of data, that kind, and basically FLAM would come up with one locus predicting the phenotypes, and it could model arbitrary patterns of dominance among alleles, including overdominance or underdominance. So the name of the game is to most efficiently estimate the phenotypic character using whatever large set of genomic data, genome-wide data, including 1 million SNPs, for example, most efficiently predict the phenotype efficiently in terms of the number of SNPs or other genomic features you're using. And this is an important stipulation because if there was no penalty function, let's say no premium for efficiency, you could of course arbitrarily fit any type of phenotypic data given the huge amount of genomic variables you input. Now in 2008, Larry Muller, together with some of the rest of us, did a numerical test, so this is entirely simulations, of how well FLAM would do if there were, in this case, 2,000 genomic features of interest. And here it refers to SNPs because he has to have an explicit model for the determination of the phenotypes across a variety of populations, where the determination of the phenotypes is shown in the bottom equation. And there are 5 types of loci that Larry Muller kept track of in these simulations. 1,960 were randomly distributed and had no relationship to the phenotype whatsoever. 10 SNPs out of the 2,000 in the simulations were differentiated in a correlated manner. That is to say, they were associated with your selection regime, for example. You're doing selection, but had no causal effect on the phenotype. And another 30 SNPs did have causal effects on the phenotype, and they were sorted out into 3 bins of 10 loci each, where they had strong, moderate, and weak effects, where the strong effects had magnitude 10 times greater than the weak effects. So basically, you've got 5 types of SNPs in this simulated 2,000 SNP set. And in these simulations that Larry did, he was simulating variable numbers of populations. And in the simplest cases, you can just think of them as half of them being selected and half of them being control populations. So what do the results look like? So these 2 graphs shown on the right are simulations of how many of each of those 5 different kinds of SNPs were picked up by FLAM and used in FLAM models. And if you look first at the bottom figure, panel B, this is a figure where you have a very good ability to estimate the average phenotype in a population. So that would arise, for example, if you estimated the phenotype from 200 or 300 individuals. And the upside down triangles show the number of neutral SNPs that are picked up in experimental evolution designs between 10 and 60 populations simulated, and they're essentially never picked up by FLAM. So neutral variation is never picked up by FLAM in this case, and in fact, the same thing is true of the upper case. The upper panel shows a less accurate estimation of the phenotype from the experimental populations. So for example, if you only measured 5 to 10 individuals for a phenotype. And by the way, in our lab, we never do that. For us, a minimum number of phenotypes that we derive from a population would be 20 to 30, and very often we're estimating phenotypes from hundreds of individuals. So for me, it's really the bottom case that's important, but I know other labs are not as obsessive compulsive as we are. The diamond results are the frequency with which FLAM picks up SNPs that are differentiated in response to the selection regime, but where for the phenotype of interest, that particular SNP has no phenotypic effect. That is to say, there's no developmental effect, if you want to use that language. It has no causal effect on the phenotype. It's just a spurious but fully correlated genomic signal. If you estimate your phenotypes accurately, the bottom panel, FLAM very rarely picks up such spuriously differentiated SNPs. If you only inaccurately estimate your phenotypes, which is shown in the top panel, FLAM will pick up these spuriously differentiated SNPs a fair amount of time, about 20% of the time at, for example, 40 populations, and then it doesn't get any worse than that. Now, the dark symbols show the success of FLAM at picking up alleles with strong, moderate, or weak effects, where you don't have any strong nonlinearity in the impact of SNP frequency changes or TE frequency changes relative to the phenotype. So crudely speaking, if you're somewhere in the roughly additive to moderately dominant range, amazingly, this particular technology, this type of machine learning, is very good at picking up strong, moderate, or weakly contributing SNPs with respect to the phenotype, where the number of such genomic features it's picking up scales upward as the number of populations you're looking at goes up. And roughly speaking, we're looking at a low ceiling on the number of SNPs that are available, because remember, there are only 10 SNPs of each kind. So if you look at the bottom panel, there are only 30 causal SNPs in the simulated set of 2,000 SNPs that FLAM could possibly pick up, and it's getting 60% of them strong, moderate, weak at 60 populations worth of data. And this is going to bend as it approaches 100 or 200 populations, approaching saturation at about 95% of the available 30 loci. Now, this is all in the world of simulations, and of course, what you're really going to be interested in is how does this do in real life? A very important feature of using FLAM, which we sort of accidentally discovered as we were using the Peterson software in R, is you have to do permutations, or FLAM will just take your data in the order you have it ordered in, which in the case of flies is a well-defined genomic order. And once it's got a good model, it then stops. So what you have to do is you have to permute your genomic information and rerun FLAM to properly sample through the entire genome. And Larry Muller decided it would be cool to take advantage of this to have a, in effect, statistical exploration of any particular body of data through 100 permutations of your genomes, which will 100 FLAM models and a different set of 100 FLAM sparse sets, meaning the subset of genomic features used to predict the results, where in order for us as a scientist to infer that a particular genomic feature or locus can play a causal role is we stipulate that it be present in 50, 40, 30, or 20% of the FLAM lists. Now, when we do that, as shown in the bottom panel, this is how many non-causal but differentiated loci are being picked up by FLAM, depending on your level of stringency. And you have to go to 50% stringency to reduce that proportion to very low levels. Those are the dark circles. In so doing, you sacrifice the number of loci you're going to detect as causally contributing. In what I'm going to be showing you going forward, we use the 50% criterion, so we believe that very rarely are we picking up spuriously correlated SNPs. I'll just say in passing, this is an important footnote, genetically linked genomic features like SNPs that are close together will be confounded with each other, and will be a non-causal linked SNP can be spuriously picked up. And however, we have a fix that we use. So here I'm going to show you some data from a preprint where we're applying FLAM to the transcriptomic results in 20 populations called A versus C, which have evolved substantial differences in their patterns of aging. These are log mortality plots for adult mortality, where typical fast cultured or early cultured flies are shown in green. They have fast aging. Their mortality rates, even on a log scale, shoot up linearly before they hit the late life plateau. The blue data, which are from the flies that were cultured at 28 days of age from egg, exhibit a classic delayed aging pattern in which there is basically no aging for the first 28 days of life, defined in terms of mortality rates. And these are both male and female data, male data on the left, female data on the right. So what we have is a very striking phenotypic difference in patterns of aging. These were genome-wide sequenced. Here you're looking at about 1 million SNP tests plotted as dots. This is a CMH test. We get qualitatively similar results from quasi-binomial tests and other tests. Here a resampling statistical threshold of about 10 to the minus 160 was used. That's the red line, which nonetheless gave us, of course, thousands of SNPs that are statistically differentiated. And that's from 10 A populations with fast aging compared to 10 C populations with slow aging. Then we looked at the differentiation of these populations with respect to transcripts. And excuse me, day 14 and 21. And from here on, we're going to focus on the day 21 data, because that's where we have a really big phenotypic difference in mortality rates. Basically, at that point in time, the C populations are in the middle of a 2-week period where they're not aging, whereas the A populations are rampantly aging at day 21. Here are the differentiated transcripts using our bar graph plots across the Drosophila melanogaster genome, which has 5 major chromosome arms, since the X is telocentric, and a tiny 4th chromosome. So there's differentiation at thousands of transcripts as measured in RNA-seq estimates of quantitative transcript magnitude. And then we applied the Lasso machine learning for the prediction of transcript level differentiation based on the SNP data genome-wide, transposable element, structural variants, and duplications. So first, this was done for each of those kinds of genomic features separately. We have 4 different sets of FLAM data. I'll just repeat. This was done with 100 permuted genomic sets of data. And we're picking the genomic sites that were picked up at least in 50% of the FLAM models. But it's very important to emphasize we do this on each transcript individually. So we're looking at FLAM having to do this many thousands of times to do the entire transcriptome. So here's an example of a FLAM result for one transcript, which is for the transcript level of the gene TKV, which stands for thick vein, or vein refers to the wings. And what you see is that 9 SNPs appeared more than half of the time across 100 FLAM models for the genomic determinants of this transcript level. So this, for those of you into gene regulation, is evidence for extensive transregulation for that particular transcript. If you invert it and ask, well, how many different transcripts can one SNP difference impact significantly when it's competing against all the other SNPs? And here's what the answer looks like. It can be in the high teens. So one genomic piece of genomic differentiation can affect many transcripts distributed all across the genome. And remember, this is going to be an underestimate with respect to all of these connections. If you then look at all the SNPs predicting the 539 statistically differentiated transcripts at day 21, and you bin your SNPs into 194 linkage-independent bins. You'd have to read the paper to get more detail on that. But basically, we're using 194 highly significant SNPs that are spaced apart in the genome but are highly differentiated. You end up with more than 3 interactions between genome and transcriptome. Now, this is the kind of number that would give your conventional reductionist molecular geneticist a heart attack. But for me, that's an exciting number because I've always been a quantitative geneticist since the early 1970s. So, hey, that looks a lot like R.A. Fisher to me. We're still right. With TE differentiation, the TEs also have a significant impact on the transcriptome. As do indels, very complicated indel pattern. Duplications, less impact. I'll also comment that as we go back and forth across this set of genomic features, the predictive success of the SNPs genome-wide averages in the 60% to around 70% predictive success, which is a high number by GWAS standards, if you do GWAS. Whereas the copy number differentiation, we only had 69 sequence duplications genome-wide. This is very weakly predictive of what's going on in the transcriptome. If you combine them all together, you get this result, which is overwhelmingly dominated by SNP differences. And for now, we think that that's an artifact of how FLAM works. So that is all stuff from a paper, which is posted in bioRxiv and is under review right now as we speak. Just very briefly, this is work in progress. I'm mentioning it because here we are working with 50 populations, genome-wide sequence. I'm only going to be showing you SNP data. These are 50 populations subject to 5 different selection protocols in groups of 10. And the characters that we're looking at now are physiological characters, what a regular biologist might consider real characters, characters like starvation resistance, desiccation resistance, which we've published on a lot over the last 35 years. Very big parts of life history, evolution, and fruit flies. We added to this data set cardiac arrest rates when you pace, which is basically the electromechanical version of feeding flies cocaine. Of course, we don't use cocaine. And the more you pace them, the faster their hearts give up, and longer live flies and have more robust hearts unless they're super fat, which are obese SCO populations, which die of heart attacks very fast, which in turn is largely predicted by average lipid content. This is why you don't want to be morbidly obese. It taxes the heart. This is true not only in mammals, it's also true in fruit flies. I'm just showing you these data to give you a feeling for the extensive differentiation across sets. This is the data organized in groups of 5 populations at a time, which have common selection histories over the last 40 years. And so extensive differentiation. Here are FLAM results, just looking at SNPs, both looking at SNP bins, which I don't have time to explain in the main body of my talk now, but also at our best candidate genes within each SNP bin, which are within a 1 kb window of our most strongly differentiated SNP within a bin. And here you see, very interestingly, despite, in the left-hand column, despite having 50 populations worth of data, FLAM is picking up 12 genes. Now that's a conservative value, but the success in predicting heart attack survival from those 12 genes is actually very high. It's not a low predictive success. It's higher than you get in the vast majority of GWAS models. Desiccation resistance, ability to survive being desiccated, 14 genes. Starvation resistance, 37 genes. So this technique can show a genetic diversity of genomic features across the genome, determining a phenotypic character, or it can zero in on a smaller subset of genes if that is more likely to be the causal pattern. That is to say, as I said earlier, if there's one gene of overwhelmingly large effect that determines a phenotype, FLAM is perfectly happy to endorse that gene and say, yeah, it's Mendelian variant of large effect. But if that's not the case, then FLAM will give you an ability to identify the most obviously contributing genetic loci. This is no guarantee there aren't. In the case of starvation resistance, for example, shown here, where we've got 37 pretty good implicated genes, that doesn't mean there aren't 100 other genes of very, very small effect. They might well be there, but those 37 genes having a very large effect on that phenotype. Which brings me to the end. So the bad old tradition of biology is to pick your favorite gene or genes and then go looking for evidence concerning what it does. Experimental evolution, followed by pool-seq and transcriptome-wide RNA-seq, gives you an unprejudiced way of characterizing relationships between the entire genome and phenotypes. Here I've illustrated the value of machine learning to do this last step objectively by definable rules. I should emphasize that it is only one of many alternative machine learning algorithms you could use. And we have in fact collaborated with AI people in the private sector who played with our data and also get sparse sets using their machine learning algorithms, which again have some type of penalty on gratuitously adding variables that contribute little. You know, my scientific summary is the genome to phenotype relationship is generically too complicated for using any non-algorithmic approach, such as picking your favorite candidate loci. Thank you.